Welcome to this lecture on evolutionary biology. Today, we are going to explore primate characteristics, which form the evolutionary foundation of our entire lineage. If you have ever wondered why humans are built the way we are, the answers lie deep in our primate past. By the end of this session, you will understand exactly what makes a primate unique and how we can mathematically measure these evolutionary adaptations. To start our journey, imagine trying to climb a tall tree, catch a fast-moving insect, and judge the exact distance to the next branch all at the exact same time. If you were a dog or a horse, this would be nearly impossible because your paws cannot grasp, and your eyes are located on the sides of your head. Primates, however, are like the ultimate biological Swiss Army knives of the forest canopy. We evolved specialized tools like grasping hands, forward-facing eyes, and highly developed brains to survive in these dense, three-dimensional environments. Think about how easily you hold a smartphone. That is your ancient primate heritage at work. Now that we have an intuitive feel for these traits, let us look at the formal biological definition. In evolutionary biology, the order primates is defined by a suite of shared derived characteristics, which we call synapomorphies. These traits primarily evolved as adaptations for an arboreal, or tree-dwelling, lifestyle. Unlike other mammals, such as cheetahs or horses that specialized in running or hunting on the ground, primates retained a generalized limb structure. This generalized structure provided the flexibility needed to move through unpredictable forest canopies, setting the stage for the development of highly specialized hands and feet. Building on that generalized limb structure, let us examine the specific physical traits that define primates. Key among these are prehensile, or grasping hands and feet equipped with opposable thumbs, allowing for a highly precise grip. Furthermore, primates possess flat nails instead of sharp claws. This evolutionary shift provides sensitive tactile pads on the fingertips, which are essential for exploring the environment safely and handling delicate food. Another defining trait is stereoscopic vision. Because primate eyes face forward, their fields of view overlap, granting the excellent depth perception required to leap accurately from branch to branch. Beyond physical traits like hands and eyes, we must also consider how primates develop and interact. Primates are characterized by a relatively large brain-to-body size ratio, which supports advanced cognitive functions. Accompanying this large brain is an extended life history. This means primates typically have longer pregnancies, give birth to fewer offspring at a time, and experience a prolonged period of childhood dependency. This extended childhood is absolutely critical. It provides the necessary time for young primates to learn complex social behaviors, survival skills, and environmental navigation from their parents and community before they have to survive on their own. Understanding these defining traits naturally leads us to ask why they matter so much to scientists today. The answer is that primate characteristics provide the evolutionary baseline for everything that makes humans unique. If we want to understand why humans have such massive brains, why we rely so heavily on vision rather than smell, or why we have such complex social structures, we must first look at our primate heritage. In the real world, these characteristics also dictate how modern primates interact with their ecosystems, such as dispersing seeds through fruit consumption or navigating fragmented forests. In addition to ecology and evolution, these shared traits have profound implications for modern medicine. Biomedical research relies heavily on understanding primate biology. Because of their genetic and physiological similarities to humans, non-human primates are critical for studying complex diseases, brain development, and genetics. Without a deep grasp of what makes a primate, the entire story of human evolution, from the first bipedal hominins to modern humans, would lack its foundational context. We study them to ultimately understand the biological machinery that operates within our own bodies. Up to this point, we have discussed primate characteristics in anatomical and behavioral terms. But how do we measure them scientifically? Evolutionary biologists and physical anthropologists use quantitative metrics to understand how these traits function in different environments. We cannot simply look at a bone and guess how an animal moved. We need rigorous, repeatable measurements. One of the most important mathematical tools used to study primate limb proportions and locomotion is the intermembral index. This index helps us mathematically determine whether a primate is adapted for leaping, walking on all fours, or swinging through trees. Now that we know what the intermembral index is used for, let us look at the actual mathematics behind it. The formula is I equals LF divided by LH, all multiplied by 100. Conceptually, this formula compares the length of the arms to the length of the legs. The variable I represents the intermembral index, which is a dimensionless percentage. The variable LF represents the total length of the forelimb, calculated by adding the humerus and radius bones. The variable LH represents the total length of the hind limb, adding the femur and tibia. If the forelimb length increases relative to the hind limb, the index rises above 100, indicating long arms built for swinging. If the hind limb increases, the index drops below 100, characteristic of leaping primates. With our formula established, let us apply it to a real-world scenario involving a lemur from Madagascar. We are given that the forelimb length is 21 centimeters and the hind limb length is 30 centimeters. We need to calculate the intermembral index and determine its locomotion. First, we substitute 21 for the forelimb and 30 for the hind limb into our equation. Dividing 21 by 30 gives us a decimal ratio of 0.70. This means the arms are exactly 70% as long as the legs. Finally, multiplying by the constant 100 gives us a final index of 70. Because this value is significantly less than 100, it mathematically proves this lemur is adapted for vertical clinging and leaping, using powerful hind limbs. Having seen a leaping primate, let us now compare a gibbon to a human to see extreme evolutionary divergence. First, we will calculate the index for the gibbon, a lesser ape. We are given a forelimb length of 65 centimeters and a hind limb length of 50 centimeters. We place 65 in the numerator and 50 in the denominator. This setup asks how much arm exists for every unit of leg. Dividing 65 by 50 yields 1.30. Multiplying by 100 gives a final index of 130. This high number mathematically demonstrates that a gibbon has arms 30% longer than its legs, which is a perfect evolutionary adaptation for swinging beneath tree branches, known as brachiation. Now that we have the Gibbon's index, let us perform the exact same calculation for a modern human. For the human, the forelimb length is 75 centimeters, and the hind limb length is 105 centimeters. Notice that, unlike the gibbon, our denominator is much larger than our numerator, reflecting our reliance on legs for transport. Dividing 75 by 105 gives approximately 0.714. Multiplying by 100 yields a final index of 71.4. This stark contrast to the gibbon highlights a major evolutionary divergence. While the gibbon evolved massive arms for the canopy, humans evolved elongated, powerful legs for efficient bipedal striding on the open savanna. To truly cement these mathematical differences, let us look at this visual comparison of primate skeletons. On the left, you see a primate with longer hind limbs, represented by the downward arrow labeled LH. This structure, as we calculated for the human and the lemur, results in a lower intermembral index, suited for terrestrial walking or leaping. On the right, you see a brachiating ape hanging from a branch. Notice how the forelimb, labeled LF, is drastically longer than the hind limb. The central formula connects these visual realities to the math we just performed. This diagram perfectly illustrates how the intermembral index captures the physical adaptations required for different evolutionary lifestyles, from the forest canopy to the ground. As we wrap up this lecture, let us review the most important ideas we have covered. First, primate characteristics represent a unique bundle of evolutionary adaptations specifically designed for life in complex, three-dimensional arboreal environments. Second, traits like grasping hands, forward-facing eyes, and large brains allowed our early ancestors to thrive and form complex social structures. Third, we learned that we can quantify these physical traits using mathematical tools like the intermembral index, which traces the evolutionary journey from tree-dwelling quadrupeds to bipedal humans. You have now covered the full foundational topic of primate characteristics, which is the first essential step in unraveling the fascinating story of human evolution.